Hey everyone, and welcome back. To all the ninth grade biology students of St. Paul American School Clark tuning in, welcome to today's Explainer. We are diving into Unit 2, Lesson 3, the plasma membrane and structures, and organelles. We're going to be looking at the microscopic world in a whole new way today, so let's get right into it. Here is our objective for today. By the end of this, you're going to understand exactly how the structure of the plasma membrane dictates its function. Specifically, how it regulates transport to maintain homeostasis. Now, what we're looking at here is pretty amazing. This isn't just some textbook drawing. It's a real transmission electron microscope image of an actual cell membrane. You can literally see the distinct layers forming the only boundary that stands between the highly organized machinery of the cell inside and the totally chaotic world on the outside. So, why does this matter? Well, this membrane isn't just sitting there like a static brick wall holding things in. It's incredibly active. Its main job is to maintain homeostasis, which is that perfect internal balance within a cell. Think of it kind of like the climate control system in a smart home, right? It's constantly monitoring, adjusting, and controlling what comes in and what goes out, so the inside stays perfectly stable, regardless of what's happening outside. And this right here perfectly illustrates the chemical foundation of that boundary. It's all about these little guys called phospholipids. A single phospholipid has a round, polar phosphate head that absolutely loves water, and it's attached to nonpolar fatty acid tails that are, well, terrified of water. So when you put a bunch of them in a watery environment, they automatically assemble themselves into a lipid bilayer. The water-loving heads face the outside, and the water-fearing tails tuck themselves safely away in the middle. When you put it all together, this builds into the complete structure we call the fluid mosaic model. It's a mosaic because that lipid bilayer is just packed with embedded molecules, things like large transport proteins and carbohydrate chains that act as little cellular identification tags. And it's fluid because these components aren't locked in place. They dynamically float and shift around. And actually, keep in mind, this exact same flexible membrane structure is also what makes up the structures and organelles inside the cell. Because of this super specific, multi-layered structure, the membrane is what we call selectively permeable. That basically means it allows some specific molecules to enter the cell while completely blocking others. It's literally acting as a highly sophisticated bouncer for the cell, checking IDs at the door and only letting the right guests inside. So how do things actually get past that bouncer? We basically have two main rules of movement or cellular transport. On one hand, we have passive transport. This takes zero energy from the cell. Molecules just coast freely from an area of high concentration down to an area of low concentration. On the flip side, we have active transport. This is where the cell actually has to spend its own energy to force molecules from an area of low concentration up into an area of high concentration. Let's look a little closer at passive transport first. The most basic form is simple diffusion. Here, molecules are just in random motion. If you've got a really high concentration of particles crowded in one corner, they're naturally going to bump into each other and spread out toward the empty space. They drift from high to low. And this keeps happening until they are completely evenly spaced out, a state we call equilibrium. The cool part, even at equilibrium, they're still moving, but they just stay perfectly balanced. But, what if a molecule is just too large? Or what if it has a chemical charge, like an ion, and just can't squeeze through those lipid tails? That is where facilitated diffusion comes into play. The membrane uses specific proteins to help out. Channel proteins act like open tunnels for specific ions, while carrier proteins actually grab a target molecule, change their own shape, and drop it on the other side. It's still passive, it still flows from high to low, but it just needs a dedicated VIP pathway. Then we have a very famous type of passive transport called osmosis. This is specifically the diffusion of water through a selectively permeable membrane. See, when solute molecules are just way too big to pass through the membrane's pores to balance things out, the water moves instead. It flows freely from where there is a high concentration of water to a low concentration of water, just trying to balance the whole system. Okay, now let's shift gears completely to active transport. Sometimes the cell has to fight the current. It needs to stockpile nutrients or push out waste against the natural flow. To move molecules from a low concentration into an area that's already highly concentrated, the cell has to spend energy. It uses specialized transport proteins called protein pumps to physically force these molecules across the boundary. Let's walk through a real-world example of this in action, the sodium-potassium pump.
This is an active transport mechanism that is absolutely crucial for your nerve responses, and it operates in 3 distinct, energy-driven steps. Step 1. Sodium ions inside the cell bind to the carrier protein, and energy in the form of ATP loses a phosphate group. Step 2. That free phosphate binds to the protein. This acts like a switch, changing the protein's entire shape. Step 3. Because of that new shape, the sodium is actively forced out of the cell.
But what about the really massive jobs? Sometimes the cell needs to move materials that are just way too big for even a protein pump. For these bulk transport jobs, the cell uses energy to literally change its own shape. In endocytosis, the membrane folds inward, bringing large materials inside by pinching off into a tiny little organelle called a vesicle. Exocytosis is the exact opposite. The cell wraps waste in an internal vesicle, merges it with the outer membrane, and shoves it all out in bulk. Now, as a fascinating analysis model, let's think about how complex these integral membrane proteins really are. They aren't just dumb transporters, they act as highly sophisticated communication receptors. In step 1, they receive extracellular inputs like hormones. In step 2, once bound, they activate complex intracellular processing cascades. But here is the wild part. Step 3. This system is so precise that some viruses like HIV have actually evolved to hijack these exact receptors to trick the cell and break inside. Actually, scratch that. Before we wrap up, it's vital we do a quick misconception check. You might hear about the old sandwich model, also known as the Davison-Danieli model. Early scientists thought the membrane was a rigid sandwich, just a simple lipid layer stuck between 2 flat, static sheets of proteins on the outside. We now know there's no way that's right. It was way too simplistic, it totally ignored membrane fluidity, and it failed to recognize the incredible diversity of proteins embedded all the way through the membrane. Okay, quick formative check to see if we're all on the same page. Based on everything we just learned about osmosis, think about how water affects the size of red blood cells. If a cell shrinks because water is leaving it, what kind of solution is it in? If it stays perfectly the same, what is that called? And if it swells up like a balloon because water is rushing in, what is that? Keep those terms, hypertonic, isotonic, and hypotonic, in mind. Let's do a really quick recap to lock this all in. First, the plasma membrane, as well as the boundaries of internal organelles, is a selectively permeable lipid bilayer, described by the fluid mosaic model. Second, passive transport moves materials from high to low concentration without using a drop of energy. Third, active transport uses cellular energy to pump materials from low to high concentration, constantly fighting to maintain that crucial homeostasis. I want to leave you with a provocative thought from our sources today. Considering just how incredibly precise this gatekeeping system is, relying on exact protein shapes to send signals and allow nutrients inside, what catastrophic chain reactions might happen in your body if the genes encoding these membrane receptors were to suddenly mutate? Think about the disastrous consequences of a malfunction in that signal transduction process. Thank you so much for joining me for this explainer, and keep exploring.